thank you very much. It's my really distinct pleasure to have the opportunity to come and to present to you all. And thank you for Claudia and the Pope for welcoming all of us here. Yes, first female Pope. It's a real pleasure. I can kiss the ring. In any case, I'm going to be not really giving you an overview of all of neuroimaging here. I'm going to be highlighting some of our more recent work into trying to understand the brain mechanisms by which acupuncture works for a unique chronic pain disorder, carpal tunnel syndrome. So the contention here is that acupuncture can really be separated into distinct components. There's the ritual, the TCM ritual associated with the therapy itself, the history taking, the palpation, diagnosis, and the therapeutic alliance that's built up between the patient and the clinician. The needles are then inserted, and this includes a somatosensory stimulation, so be it mechanical of the needle itself, thermal if you're using moxa, and or even electrical stimulation if you're doing electroacupuncture. And the needles are left in place, and there's a relaxation response that happens. And it's very likely that the mechanisms of acupuncture are probably distinct between these different components of the therapy itself. And we have to be mindful that when we're studying mechanisms of acupuncture, we really have to be specific as to what aspect, what component of acupuncture we're most interested in. In this case, I'm going to be focusing on, during this talk, on the somatosensory stimulation aspect of acupuncture, specifically electroacupuncture, and we're going to be focusing on a part of the brain called the somatosensory cortex. So we decided to focus our study on carpal tunnel syndrome, and I'll tell you a little bit about it. CTS, or carpal tunnel syndrome, is the most common entrapment neuropathy. This is entrapment of the median nerve as it courses through the carpal tunnel. And the median nerve becomes entrapped. There's an increase in pressure in the carpal tunnel, and this is the vicious cycle of CTS. The increase in pressure leads to ischemia and inflammation, which damages the nerve microvasculature, leading to exudative edema and fibrosis in this area, which then cycles back, increases the pressure even more, and you have this unfortunate vicious cycle that CTS patients sometimes find themselves in. All of this leads to pain and paresthesias in the fingers that are innervated by the median nerve, right? So it's a very specific lesion, a specific condition, CTS, okay? And so the question is how can acupuncture break this cycle? Can acupuncture break this cycle? So one unique aspect of CTS, and one of the reasons we decided to study it for our neuroimaging research, is that as opposed to low back pain and fibromyalgia, CTS is a chronic neuropathic pain disorder that has a very specific objective outcome associated with it. In this case, nerve conduction studies can pick up the speed of an impulse that's propagated proximally to the carpal tunnel and then across to distal pickups on median nerve innervated fingers, the first through the fourth. And CTS has been graded as mild or moderate depending on the speed of this impulse. So there's a slowing of the impulse over the median nerve in carpal tunnel syndrome. And this can be separated from severe CTS, which includes these delays, but also reduction of motor amplitudes that can sometimes lead to atrophy of the thenar eminence. This is a very more dangerous condition, and these patients are set off to surgery. And so what we're focusing on here is the mild and moderate CTS patients that we hope we can keep from progressing to severe CTS or even to recover some of their function. And so one nice thing from a research standpoint is that because we have this objective outcome, we can study both objective and subjective measures in this chronic pain population, which is something you cannot do in chronic low back pain and fibromyalgia. So here we're focusing on a part of the brain called the primary somatosensory cortex. This was first mapped by Penfield back in the 1930s. He is a surgeon from Canada, from Montreal, and he performed surgery on epileptics. These surgeries involve patients being awake during the surgery, and he went in and he stimulated with an electrode different parts of the primary somatosensory cortex, which is over here in the parietal lobule, and was able to actually ask the patients where they felt sensations when he stimulated different parts of the cortex, S1. And by doing this, he was able to map out what's called the homunculus sometimes, or the cortical representation of different body areas, somatotopy, in S1. And you can see here that there's the face, the hand, the torso, the knee, the leg. And as you can see here, it's not equally distributed. We are much more sensitive, for example, in our lips, in our hands, than we are in our back. And so we need many more neurons to process that information. And that's why you have this distorted body map associated with the homunculus in S1. We just have much larger areas, say, for the hand and the face than we do, for the back or the kneecap. And here we're focusing, of course, on the hand area of S1. And what I'd like to sort of confer to you is that we can actually map these areas. We don't have to perform surgery. We don't have to open up and zap people's brains. We can do this non-invasively with functional MRI. And so this, for example, is a subject being stimulated on the second finger, digit number 2, digit number 3, and digit number 5. As you can see here in this example, as we move from digit 2 to 3 to 5, if you imagine a center of mass, you can see it moving up the gyrus, right? So it's going up the postcentral gyrus, or S1, and this is exactly what Penfield showed over here as you go up the gyrus from thumb to index to middle to ring to little finger as you move up the gyrus over here. So we can non-invasively look at this with functional MRI. And what we found, and this was one of the first, I think it was the first study, functional MRI study in carpal tunnel syndrome patients, is that compared to healthy adults, where the center of masses of their representations are nicely separated between digits 2 and 3 and 5, in carpal tunnel syndrome patients, there's a contraction, a blurring of the fingers innervated by the median nerve, digit number 2 and digit number 3, where still digit number 5, the ulnar nerve innervated pinky, is still nicely separated even in this patient population. And then what we did in this was our pilot study, by the way, so that's why the ends are very small. We sent off these patients to a semi-individualized protocol, 3 times acupuncture for 3 weeks, 2 times for 2 weeks, where every single patient receives some common stimulation, common points. This is electroacupuncture low frequency between triple warmer 5 and pericardium 7. And then acupuncturists could choose manual acupuncture at 3 of 6 acupoints chosen depending on each individual subject presentation. This was kind of our way to both be precise from a research standpoint, but also try to be reflective of the clinical practice of acupuncture itself, where each individual patient might get different protocols, different acupoints being stimulated. And so what we found is that after, this is all much older research, and you can see here, there was improvement in paresthesias, for example, and pain. There was improvement in median nerve conduction latencies, but also we saw a remapping in the brain. So this is a retest from the healthy adults, again showing nice separation between digits 2 and 3 and 5. And in the patients, we got better separation between the median nerve innervated fingers in conjunction with improvements at the wrist of the nerve and in the sensations that they feel, the subjective pain and paresthesias that they feel. So our model for this was that the pain and the paresthesias that the patients are feeling lead to more frequent and abnormal nerve traffic to the brain, which then through disinhibitory processes leads to more extensive and wider digit representations in S1. But it's more than this. The paresthesias that the patients feel, and if you're a clinician and you've seen these patients, you know that the paresthesias are diffuse. They're usually not in any one individual finger. They're spread out over the fingers innervated by the median nerve. And this leads to more simultaneous signaling from affective digits. And then through Hebbian plasticity mechanisms, a neuroscientific way that synapses get rearranged and the brain gets rewired, leads to less separated and blurred digit representations in the primary somatosensory cortex. So they don't just get wider, but they actually come closer together, they blur, and this is what we think was happening in CTS patients. So then we followed this up with a larger study. We were able to get a grant to do a larger study. In this case, it was sham controlled, and we even added an interesting a third group of distal acupuncture. So there was 3 groups: local acupuncture, distal acupuncture, where the patients are stimulated on the opposite leg, on the opposite foot, opposite ankle to where the most affected hand is, and also a sham acupuncture condition with Streitberger needles. These non-insertive needles was used as a control, still with the electroacupuncture device, but without actually turning the electricity on. Patients were evaluated both at baseline with fMRI, and then following 8 weeks of acupuncture, evaluated again. So we went a little bit longer this time. We went 8 weeks of acupuncture. And we had a long-term follow-up, a 3-month follow-up with just the Boston Carpal Tunnel Syndrome questionnaire. This is the pain and paresthesia scores. This is the subjective outcome that we were able to get as a long-term follow-up. So the distal acupuncture style, of course I get this question a lot, why do we choose this? Well, first of all, it's a legitimate form of acupuncture that's practiced. There's a Tan style and Tong style, these different styles of acupuncture, where you actually would be doing this type of acupuncture. That's one case. So we wanted it, first of all, as a control for local effects. We know that there's axon reflex, there's flare, there's all kinds of effects of just sticking a needle into the location in terms of blood flow reorganization. So what happens if we have real acupuncture, but not right at the site of the lesion, right? So the lesion is in a discrete place. What if we stimulate with real acupuncture, but not at the site of the lesion? And so in this case, in some of these styles, if you have for example, shoulder pain, you might treat the hip on the opposite side of the body. If you have wrist pain, well, you might treat the ankle on the opposite side of the body. And this is, in fact, what we did here. What is the neurophysiological basis of some of these treatments? It's really never been evaluated. Interesting ideas could be the fact that, we're bipedal organisms. When we walk, when we run, there's a synchrony between the leg and the opposite arm as we transverse. And so there might be some sort of mapping that happens through development, through evolutionarily over time. You have these interesting links between opposite-sided limbs that could develop over time. This is purely speculation. So there was 80 patients that were originally enrolled. 70 of them were randomized to a local distal or sham acupuncture group. And once we got them into the study, we did pretty well. 66 of them we were able to retain in the study and came back for evaluation with fMRI and other MRI measures in the local distal and sham acupuncture group. And we got a decent amount at the 3-month follow-up as well. 55 of them completed their questionnaires for the subjective outcomes. So what we found is that, first of all, we were able to replicate our original result, which was that patients with carpal tunnel syndrome have contracted or less separated D2, D3 separation distance. So the second and the third finger are mapped closer together in S1 in the brain. So this is something that replicated from the previous finding, which is important because, first of all, it's a much larger sample size, and it's also a completely different cohort of patients. This was replicated both with functional MRI and with magnetoencephalography, which was another technique that we used to evaluate these patients, which is also nice and supportive. And interestingly, we also wanted to know what is the consequence of this remapping in the brain. So is it just, for example, an epiphenomenon? Could this be something that's downstream of the effect, doesn't really relate to the clinical picture, or is this something that relates to some of the deficits that these patients feel? And so one of the deficits that they could feel is, first of all, is fine motor skills. And while we didn't, in this particular case, we didn't look at a particular functional task with fine motor skills, we looked at a phenomenon called finger agnosia, meaning if you blindfold the patient and you stimulate one of their fingers and you ask them which finger was stimulated, we predicted that the patients would make more mistakes than healthy controls because it's the blurring in S1 that differentiates different fingers, okay? And we found that, in fact, to be the case, that as opposed to the ulnar nerve innervated pinky, where there was not a difference in correct responses between healthy controls and patients, in the median nerve innervated D2 and D3, the patients did in fact make more mistakes. And what was even more interesting was that there was a correlation between the D2, D3 separation distance and the mistakes that they made. So those who had the most contraction between D2, D3 in the brain, in the primary somatosensory cortex, those were the patients who made the most mistakes on this test, suggesting that this altered S1 somatotopy is indeed maladaptive, right? It's not just the coping mechanisms that the patients are using to try to respond to their deficit, but it's actually tracking the amount of mistakes that they make, okay? So this is now the longitudinal data that we have from the trial itself, and this was recently published. So first of all, statistically, we found no difference between the local and the distal acupuncture group for baseline versus post-acupuncture. And so the results were combined into a single Verum acupuncture group. Again, I reiterate, this is just for the pain and paresthesias that the patients are feeling. There was no difference between Verum and sham also following therapy. You can see that over here. There's really nothing. There's nothing going on here. There's no difference in pain and paresthesia reporting. Granted, as a clinical trial, this is a very small trial, so keep that in mind. But we saw no differences here whatsoever. However, if we looked at the 3-month follow-up, there was, in fact, retention of the improvement for the verum group that was not there for the sham group. And this is the breakdown of the local, distal, and sham acupuncture groups. Again, if you wanted to see it, but similarly showing that all 3 groups are improving about the same amount. So this was very different from the situation that we saw with median nerve conduction latency. This is the speed of the impulse over the carpal tunnel. In this case, so what happens is that in patients, the speed is slowed, right? So the latency is longer. So an improvement is a reduction in the latency, and that's in fact what we saw with verum acupuncture. And in sham acupuncture, it actually got a little bit worse over time. So the sham acupuncture, the latency was increased, and there was a statistically significant difference between verum and sham here. If we break it down into local, distal, and sham groups, there was a significant difference between both local acupuncture and the sham group and distal acupuncture in the sham group. So somehow, both needling directly at the lesion and needling on the opposite leg, on the opposite side of the body, also improved the nerve conduction latencies, which was interesting. We also looked at, of course, the remapping in the brain. And here we saw for verum acupuncture, there was an increase in the separation distance in S1. This was not seen for sham acupuncture. And here, when we break it down into local, distal, and sham groups, here we found a significant improvement strictly for local versus sham, although you could see it's just a smaller effect size for distal. It does look to be kind of midway between the local and the sham groups. So finally, and really interestingly, we found that there was a connection between the remapping that they have in the brain immediately after the 8 weeks of acupuncture and long-term outcomes. So those patients who increased their D2, D3 distance the most after the acupuncture were the patients who had the most retention of improvement, retention of reduction in pain and paresthesia effects at the 3-month outcome. So there's a consequence of successfully remapping the brain in these patients. It leads to better long-term clinical outcomes, because at the end of the day, patients are not coming to us complaining about remapping in the brain and the fact that their D2 and D3 is blurred, right? They're coming to us complaining about the pain and paresthesias, the symptoms that they feel as a result of carpal tunnel syndrome. But what's interesting is that these objective neurophysiological metrics are connected to the subjective outcomes. And so it's important to track these things, especially for a disorder like carpal tunnel syndrome, where we have these objective markers that can be tracked. So our model for what's happening is that while local acupuncture could work by both a direct effect directly at the lesion itself, because this is where we're doing the needling and the electroacupuncture stimulation, there's also an indirect effect that could be operating by the local acupuncture as well, because we're also stimulating receptors, sensory receptors at this local area, and by stimulating the brain, we can also then induce through, not directly through connections from the somatosensory cortex, probably through connection between the somatosensory cortex and perhaps autonomic control areas, and Johannes is going to be talking about this in the next talk, that there are these autonomic centers that can control localized blood flow in different parts of the body. So potentially, this is a mechanism by which somatosensory remodeling can affect some sort of remodeling locally in the body at the wrist. Distal acupuncture, on the other hand, does not have this local mechanism. It purely must function through an indirect mechanism through the brain, okay? And so that's why it's so interesting to track this. And that's one of our future directions, is not just to do that and to track some of these autonomic sequelae, but also to determine the temporal order of local and brain plasticity, what's driving what. Is the brain plasticity a downstream marker of something, a change that's happening at the wrist, or can it actually be used to drive changes that are happening at the wrist? We don't know the temporal order of some of these changes between nerve conduction latencies and brain remodeling. So again, just to summarize, probably in CTS, irregular polydigit signaling, as you can see here, this is traffic over the median nerve, is leading to this reduced separation between D2 and D3. Then when you impart the electroacupuncture, it's a much larger, this is 2 Hz stimulation, much larger impulses, but it's very regulated. It's boom, boom, boom at 2 Hz. And what we think that's happening is that this is regulating traffic now in the brain, and because of the regulated nature of it, is then remapping the cortical representations in S1. So the take-home message here is that while both Verum and sham acupuncture improve subjective psychological outcomes, only Verum acupuncture improved objective physiological outcomes such as nerve conduction latency and D2, D3 cortical separation. And it's, in some ways, I think it's really a shame that we don't have these objective outcomes for low back pain, even migraine, some of these other much more common chronic pain disorders, because we might be in a very different situation now in terms of reimbursement, in terms of acceptance, if we had these objective outcomes for low back pain as we do for something like carpal tunnel syndrome. So one of the call to arms, of course, is to try to develop objective outcomes as a better diagnostic for low back pain and some of these other more common chronic pain disorders. Also, acupuncture, particularly electroacupuncture, is in my mind a somatosensory guided neuromodulatory therapy that I think works maybe in some way similarly to other peripheral neuromodulation type therapies that are becoming much more popular right now. I don't know about Switzerland, but neuromodulation is becoming, such as TDCS, TMS. These are types of neuromodulatory therapies that are working directly on the brain, but there's also peripherally directed neuromodulatory therapies, so-called electroceuticals, that a term coined, of course, by the pharmaceutical industry, which is now trying to dive in and take control of this as well. But let's not get on that. Interestingly, also, local versus distal acupuncture might improve peripheral nerve function by somatotopically distinct S1 neuroplasticity. I didn't have enough time to dive too much deeply into that, but you can read about it in the paper. And the question here is, could this potentially be a basis for acupoint specificity? So S1 is one of the few areas in the brain where you do have distinct mapping for distinct body areas. So one of the hallmarks of acupuncture, of course, are acupoints, right? You have these distinct effects, distinct clinical effects that are happening if you stimulate different parts of the body. It's very possible that S1 might be sort of the gatekeeper between how stimulation at different body areas then gets transmitted to other different effector areas and emotion processing areas and things like this. And so that's something that we are actively engaged in researching right now. So thank you very much for your attention. And of course, I need to acknowledge the generous support of our sponsors, particularly the NIH, and also a slew of collaborators who have just been excellent, particularly Yumi Maeda, who was the main postdoc that was running the secondary study, the follow-up R1 funded study that we did in these patients. Joe Adet as a senior designer for a lot of this work. I was actually, the data that I just showed you literally took over 15 years as a process, from collecting the pilot study to publishing the pilot papers to applying for the grants, getting rejected, applying again, getting the grant, doing the follow-up study, and then finally these papers are coming out in 2017. So it's been a long road, and I was actually hired as a postdoc on the original pilot study to come in. So it's a long road for neuroimaging clinical research. Thank you for your attention. I'm happy to answer any questions.